Today's webinar is titled Innovative Remediation Options and Molecular Biological Tools for Performance Monitoring. And we have three speakers today. First will be myself. My name is Brad Elkins. I'm with EOS Remediation. Following my talk will be Dora Taggart. She is the president of Microbial Insights based out of Tennessee. And Dora received a biomedical engineering degree from Vanderbilt University. She joined Microbial Insights in 2001. And since then, she's been directly involved in commercializing over 60 different nucleic acid-based analyses. And under her direction, Microbial Insights has become a worldwide provider for molecular tools. Following her talk and finishing us off this afternoon will be Dr. Kerry Sublette. He is the principal scientist with Bio-Enhance, but he is also a professor of chemical engineering and geosciences at the University of Tulsa. He has over 25 years of experience in bioengineering and soil remediation. He has 13 patents and 117 publications. He also chairs the International Petroleum Environmental Conference, as well as serving on the Oklahoma Board for Environmental Quality. So it is a delight to have these two distinguished presenters with me today. So without further ado, I'm going to go ahead and get started with my presentation. And mine will focus mainly on just some introductory material, kind of some overview on general bioremediation, as well as some performance metrics to consider. And that will lead us into discussing some molecular biological tools that then Dora will take it from there. So I recognize that many of the folks listening in today are already pretty educated on what bioremediation is, but just for a review and just to be sure that we're all on the same page, when we talk about bioremediation, we're really talking about providing two things in the subsurface, an electron donor and an electron acceptor. And in the case of EOS, we're providing that electron donor in the form of emulsified soybean oil, an oil emulsion. And that emulsion goes through fermentation after it's injected. That fermentation produces volatile fatty acids and hydrogen. And it's hydrogen that's used through respiration by various dechlorinating microorganisms to exchange that chlorine with hydrogen, thus degrading the contaminant. And we want this process to continue from, in this case, from PCE all the way to ethene. And each time this is going through a very stepwise process with exchanging one chlorine with a hydrogen at a time. And in the literature, this is referred to as reductive dechlorination. We here at EOS, and again, I think everyone here sort of knows this, but we kind of make this equation happen, whether that's in the subsurface or in a microcosm, or as Kerry will explain during his talk in the Bio-Enhance reactors, the fundamentals are all the same. You need to supply a donor, and then the contaminant is the electron acceptor. Kind of getting a little off course here, but just for a very brief moment, but I want to discuss this because I think it's growing in popularity over the last few years in our industry, and that's sort of combining the reductive dechlorination with an abiotic dechlorination using like a zero valent iron. And the reactions are a little bit different. So I just want to cover that because I know that I've had some folks that I've talked with get a little confused about exactly what's going on and how these technologies work in tandem. So with zero valent iron, it's a chemical process, and that is the zero valent iron actually has to touch the contaminant of concern. And in this case, we're talking about PCE. So when zero valent iron comes into contact with PCE, it can it can basically remove two chlorines from that molecule and degrade it immediately to dichloroacetylene. If this were to happen where one chlorine from each side of the carbons was removed, that would be the beta elimination. If they were from the same carbons, that would be alpha elimination. But either way, the end goal is the same. And then that, of course, oxidizes the iron to a ferrous iron species. Now, the other thing that happens in the aquifer is zero valent iron is also coming into contact with water, and there are really two things that can happen. You can get oxidation of the iron and production of hydrogen, and then that hydrogen can be used through a biological process to reductively dechlorinate the contaminant. Simultaneously, what can happen is the zero valent iron can come into contact with the contaminant, and through hydrogenolysis will remove one chlorine at a time, very similar to the traditional reductive dechlorination process. So I just point this out because folks seem to be combining these technologies quite frequently these days, and so there's a lot that can be going on when you introduce something like a zero valent iron with an emulsified oil. So finally, talking about bioremediation of petroleum. So just like chlorinated solvents, we still need both the electron donor and the acceptor, only this time the contaminant is taking on the role of the electron donor, and it is the electron acceptor now that is the limiting factor in what needs to be added to the subsurface to stimulate the process. And two of the most common electron acceptors that are used are oxygen in the form of like a calcium peroxide or some other similar oxygen compound, oxygen releasing compound that might be out there. And the other one is sulfate. And I think the big difference between these two is, you know, injectability, but also what sort of redox condition you find the site in when you are ready to address these contaminants. And I'll talk about that a little bit more in a few slides. Just real quick, some selection factors to consider. When it comes to electron donors, there's a lot to choose from, everything from lactates and molasses to emulsified oils to ZVI, as I mentioned. With electron acceptors, you know, the two big ones being oxygen and sulfate, but folks do from time to time use nitrate. I think there's sort of four main criteria that I think practitioners need to consider when they're trying to select a substrate, and that is, what is the oxidation reduction potential of the aquifer during characterization and as you're moving towards remediation. The other is, in terms of electron donors, is how much hydrogen is that substrate producing versus the amount of acidity that's produced during fermentation. Obviously, the rate at which these different materials are utilized by different microorganisms is going to have an impact on their longevity and also an impact on your schedule and scope of the project, how frequently you're going to have to go back out and re-inject. And of course, that's going to affect your bottom line. And then finally, the ease of injection and distribution. Obviously, if you have a really tight formation, something that's predominantly clay, you may want to think about using something maybe more soluble or something that's going to be much easier to inject, or maybe even considering your injection methodology altogether. And that can be a whole nother webinar. So a couple things real quick that we know about soybean oil, which is the main component of our EOS product, is that it's a triglyceride with a glycerol holding on to 3 long-chain fatty acids. And during injection, that molecule is broken down, releasing that glycerol, and then those long-chain fatty acids break apart. When that occurs, we see kind of a spike in TOC. And so what we see both in the laboratory and in the field is immediately after injection, you see this big spike in TOC, and then it kind of dies off very quickly. One explanation for that is that during that hydrolysis of the triglyceride, you are releasing glycerin, and that is probably what you're seeing mostly in those initial sampling events. And then as that material is used, and it's used very rapidly, that's why you see that abrupt fall in your total organic carbon. The rest of the carbon through the rest of the life of the substrate is probably those shorter chain volatile fatty acids that are being cleaved off or fermented from the long chain fatty acids. About ORP, so like I said with the petroleum, one of the most important things to consider is, you know, where you are in the redox ladder. And one thing that I like to point out to people is that as you are reducing, you're dropping ORP, and there's a very stepwise process in which the microorganisms want to utilize different electron acceptors. One thing to keep in mind is that just because your ORP is positive doesn't mean that you're automatically in an aerobic environment. you can actually completely deoxygenate an environment and still have an ORP that is, plus 100. So I always tell people to rely on both their field measurements like DO and ORP, but also to test for things like nitrate, iron, and sulfate to find out where on this redox ladder your site is. Finally, this is a good segue slide into Dora's presentation, is why understanding your microbiology is so important. All these things that we just talked about are very dependent on having the correct microorganisms. So one example on the left-hand side of the screen is a microcosm study that was done to compare bio-augmentation with biostimulation only. So the C plot down there at the bottom is just an injection of lactate. And the one in the middle, B, is bio-augmentation and lactate. And what you can see is the shaded blocks represent the same time interval for this study. And you can see that in the same time interval, when you have both the electron donor and the right microorganisms, you get very fast results, complete degradation of the contamination. On the right-hand side of the screen is a field demonstration, and this is where the green line that I just popped up there, that's when they did their electron donor injection. The green line is when they did their bio-augmentation, and you can see that the results soon thereafter were ideal. They got reductions of cis, production of ethene, and the project has moved forward from there. So with that, I'm going to turn it over to Dora to take us from how do you understand your microbiology and how can that impact your project.
All right. Thank you very much, Brad. So as Brad mentioned, what I'm going to do hopefully here is kind of do a segue into what Kerry's going to be talking about and how we can use some of these in situ bioreactors to really help us, take the products that Brad just described to you and be able to use them for in-well treatment. And there are tools that we can use along with these bioreactors, just like we do on regular sites for monitoring, that can help us understand what's happening with those microorganisms. Are we effectively stimulating the right microbial populations to get those degradation processes that Brad was just describing for us? So today what I want to do is I want you to think, and if you've heard me present before, you've heard me say this a million times, we really want to think about the question that we want to answer at our site. So if we put one of these in situ bioreactors in so that we can get complete reductive dechlorination, we might have questions about what's the concentration of contaminant degraders that we have? Are we stimulating our dihalococoides with this treatment, not only in the treatment well, but are we also stimulating those down gradient locations? So what I want to introduce for you today, for those of you that may not be familiar with it, is a quick overview of how quantitative PCR works. That's going to give us the concentration of these organisms, and also another technology called QuantArray that can give us a real breadth of knowledge about the different microorganisms present in our system. And then I'm also going to, and then Kerry's going to show you some quantitative PCR data from some of the in situ bioreactor studies that we've done, and this can be applied whether it's petroleum hydrocarbons and we're looking for different functional genes, or for reductive dechlorination with organisms like the dihalococoides that I just mentioned. The other thing that I'm going to introduce and touch on today, just in case anyone has questions about this, because I think it's really a very effective tool to use with these in situ bioreactors, is called stable isotope probing. And this is a way that we can prove a compound that's used as a carbon and energy source by the microorganisms. We can take that compound and prove that it's biodegrading. So it's a very exciting tool. There are a lot of other molecular tools that are out there, and due to time today, obviously I'm not going to be able to touch on them, but if you have questions, feel free to contact us. So for those of you that may not be familiar, just to get everybody on the same page, qPCR and QuantArray, the basics of how this works is we have DNA that's present in our microorganisms, and every cell has DNA present that tells who that cell is, or it could be a functional gene that can help that organism to do a certain process. So what qPCR does is it takes short segments of DNA that are called primers and probes, and it goes in and it targets that particular gene that we're interested in and makes a lot of copies of it. We can then correlate that back to known concentrations, so we can see not only do we have yes or no that organism is present, but we can get a very accurate number of how many of that particular organism or that functional gene that's present in our sample. So we can see in these in situ bioreactors, do we have dihalococoides? And if we add one of these electron donors, are we stimulating and growing much larger populations of these organisms for treatment within the reactors? So as Brad was saying, it gives us that direct line of evidence of what's happening with the degradation. And there's two types of genes that I just mentioned that we can look at. We can do a taxonomic identification that will tell us who a microorganism is. Very useful in sites like for reductive dechlorination when we're talking about dihalococoides, for instance. We can also look at target genes, like functional genes for degradation. So that may be for if we have petroleum hydrocarbons at a site. And I think Kerry's going to show you an example of this later, where we can look at some of the function to see, do we have oxygenase or reductase genes that are present that can do that degradation very effectively for us. So qPCR has been around for more than a decade now. We've used it in environmental remediation. It's used in all kinds of industries, and we just transitioned it to bioremediation back in around 2000, 2001. And we know from a lot of studies that this is the most accurate way that we can identify and to get concentrations of our microorganisms. The limitation with qPCR is when we do that technology, we're looking at one organism or one functional gene at a time. So one of the things that we wanted to do was to have a way to be able to look at a lot of genes. Like Brad mentioned earlier, sometimes we're interested not only in reductive dechlorination, we may be interested in co-metabolism or other processes, or what if we have mixed contaminants at the site, like chlorinated ethenes and ethanes? There could be a lot of organisms and functional genes that we want to look at. So QuantArray is qPCR, but it's kind of qPCR, I think of it like qPCR on steroids. It's where we can extract our DNA or our RNA from our microorganisms, and rather than doing a single amplification, we can put it on an array slide like I'm showing here in this picture, and each of these squares is a sample, and each of these squares has little through holes. So the qPCR amplification happens in each of these holes and generates curves that we can correlate back to get the concentration of that organism or that functional gene. So rather than getting one number, we get back 20 to 30 different numbers that we can use, and it can be for a lot of processes. Or like for petroleum hydrocarbons, we can look at both aerobic and anaerobic degradation of BTEX and PAHs, MTBE, kind of all at the same time. So it gives us a lot of information, but it is still the same thing as qPCR. It's just more qPCR. So one thing that Kerry's going to mention today that I just wanted to touch on is I've been talking to you a lot about when a cell has DNA and we're targeting and extracting that DNA with our qPCR, it tells us the number of that organism or the number of that functional genes. So that gives us the genetic potential of what can happen at our site. It's that permanent hard copy that a cell has of that particular gene that we're targeting. Well, in nature, what Mother Nature has done to conserve energy is we have where the DNA is induced because of some activity that happens, and the DNA is transcribed to RNA, and we can also extract that RNA, which is single-stranded and short-lived and only made when the cell needs it for that particular activity, whether it's growing and making new biomass, or if it's performing a function. For instance, it's induced by a naphthalene molecule that comes by and it wants to degrade that naphthalene, so you get production of this RNA. We can extract that RNA, so it's taking a step further than where we know from DNA where that permanent hard copy is there. And RNA is showing us that the organism is actually expressing that particular activity that we're interested in and making RNA. And since that degrades very rapidly, we're not going to detect high concentrations of that unless an activity is probably less likely that activity is happening at the site. The other thing that happens just to complete the process is that RNA is translated into a protein, and that is what interacts directly most times with the molecules that we're interested in for degradation processes. Eventually, we'd like to get to this end where we can do proteomics on a commercial basis and be able to look at that and translate that to activity at a site. But for now, when we look at the RNA, that gets us a step closer to where we want to be, and we can get a lot of information that I think Kerry's going to show you the value of looking at the RNA, especially when it comes to petroleum hydrocarbons or organisms that are more ubiquitous in the environment. We know that this organism is there and it has a lot of metabolic capabilities. Now we can take it a step further and know, is that organism actually making a lot of copies of that particular gene because they're actively expressing that function. Okay, so the final tool before I turn it over to Kerry, I want to talk briefly about something called stable isotope probing. Many of you are probably familiar with this. We know that in nature, most of the compounds are a 12 carbon. So stable carbon 12 that's present, 99% of the carbon out there. Well, in this process, what we're going to do is flip the ratio, and we're going to make a compound, whether it's a 13C benzene, a 13C naphthalene, whatever the compound may be, and we're going to manufacture that compound so that it's the other stable isotope, a 13 carbon isotope, and then we can use that as a tracer to help us prove if biodegradation is occurring. So the way that this works is we would take our 13 carbon compound that was synthesized for us, load that onto the BioTrap samplers. I'm going to touch on those briefly, and then Kerry's probably going to go a lot more in depth because the beads in these BioTrap samplers are the real key to the BioTrap. It's called a Bio-Sep bead. We're going to load our compound onto these beads and put them into a PVC housing, put them in the monitoring well for a period of 30 to 60 days, and then do some analysis after. So biotraps are really interesting tools that were developed by the University of Tulsa and DuPont, and they give us a way that we can look at our microorganisms. Each of these little circles that you see here are the Bio-Sep beads, and they're about the size of a BB. These are made of powdered activated carbon and Nomex. Nomex is what a fire retardant suit's made out of. So the powder activated carbon gives us the ability to absorb compounds really well onto them. The Nomex gives us the ability to sterilize them, so we know that there's no fossil biomarkers. So what we're going to do is take these beads, and it's what's inside the in situ bioreactors that Kerry's going to show you in a minute. We're going to put those down into the bioreactor or into our monitoring well. The organisms are going to colonize there, start to utilize that compound that we've loaded on, or maybe we don't have a compound on there and they're just absorbing compounds that are present in that well. The organisms are going to start to utilize those compounds as a carbon and energy source, and then we can see what happens and what organisms are present. In the case of SIP, we've put a compound on the bead, put it down in the bioreactor, and then we're looking at what happens with those microorganisms. So we know how much loss we have of that compound that we loaded, and then we can look for that 13 carbon, since it's not commonly found in nature, into our mineralization processes or into our metabolic processes. We can look at the lipid membrane of our cells. If they're growing new biomass and making more organisms, then you're going to see that 13 carbon incorporated into their cellular structure. So then we can prove definitively that that carbon compound that we put down in that ISBR is being utilized by the microorganisms. This is just a real quick example of the data from that. This is where we took a 13 carbon naphthalene, put it onto one of the biotraps, and put it down into a location. In nature, we would see dissolved inorganic carbon would be amount of minus 25 delta value. That's our units for these, and we can talk about these if anyone has questions. But look at this, our 13 carbon naphthalene sample is showing delta values of around 200, 250. There's no way that this would happen in our location unless the organisms were mineralizing that compound and making carbon dioxide that was heavy weighted. The same thing with our biomass. As I said, they'll build new cellular structures, and if we see positive incorporation del values of this magnitude, it's indicative that the organism utilized that carbon from their naphthalene to make new biomass. So it's a way we can prove positive that the compound is biodegrading. So two tools that I think are very useful with the in situ bioreactors, just to sum it up, quantitative PCR and qPCR that or QuantArray that I talked about are ways that we can accurately quantify different contaminant degraders that we're interested in, whether we're looking at the name of an organism or whether we're looking at a particular function that we're interested in. QuantArray is the same thing as qPCR. It's just more cost effective to be able to look at multiple pathways or mixed contaminants at a site. And then finally, stable isotope probing that I just briefly touched on with you today is a way that we can collusively show that biodegradation is happening when it's something that the organism's using as a carbon and energy source. We've done this for a lot of compounds. It's a great tool for emerging contaminants because we don't have to know the microorganisms or the pathways. We're just looking at biomass as a whole being incorporated, and it can be pretty inexpensive depending on the size of the molecule that we're having synthesized. So it's a great tool that we can use. So I'm going to go ahead and sum up that on the tools. I think Kerry's going to show you some data from these. So I'll turn it over to you, Kerry.
Thank you very much, Dora. What I'd like to talk to you about today, guys, is a new in-well treatment system that we're calling an in situ bioreactor. And the purpose of this Bio-Sep in situ bioreactor is multifaceted. But first of all, the intent is just to try to make localized bioremediation as easy as possible on the practitioner. But at the same time, the ISBR does have the capability to overcome a couple of common limitations of bioremediation in groundwater that you're probably familiar with. That first limitation has to do with low contaminant concentrations. We know that there is a certain minimum threshold concentration of a biodegradable substrate that must be present in the aquifer to support biodegradation or bioremediation and growth of the microorganisms. Of course, the organisms are only going to be degrading these compounds because they can use them for food for growth. So if the thermodynamics is not there for that concentration of substrate to actually support growth, then we have persistent low contaminant concentrations. This is a situation that you're probably familiar with. Another potential limitation of biodegradation has to do with substrate inhibition. If we have high concentrations of contaminants in an aquifer that may be biodegradable, but may also be toxic to those same organisms if the concentration is too high. And what I'm going to show you is, with a couple of case studies, is how we can overcome some of these limitations, the low contaminant concentration and substrate inhibition. Now, Dora was talking to you a few moments ago about the biotrap, and as Dora said, the biotrap is basically a housing similar to the one that you see here, the one that Dora showed, that is filled with Bio-Sep beads. And as Dora said, they're made out of Nomex and powdered activated carbon. This is a cross-section of one here. So basically, you can think about this as powdered activated carbon held in a three-dimensional porous network. There's a very high degree of porosity here. About 75% of the inside of the Bio-Sep bead is empty space where microorganisms can grow. And you see in the diagram on the right, or the picture on the right, this is a picture of a biofilm on the inside of Bio-Sep beads. And now, as Dora was mentioning a few moments ago, one of the characteristics of the Bio-Sep bead is that it absorbed its surface from the powdered activated carbon and the very high surface area on the inside of the bead. And these two properties combined have, we've demonstrated repeatedly, result in rapid colonization of these beads when they're put in a native environment. So lots and lots of experience with these biotraps. Some of you may be familiar with these, may have used these in the past. They've been used all over the world for well over a decade. And as I said, we know that microorganisms love to grow in these biotraps or in the Bio-Sep beads, and they're very rapidly colonized. So these observations have led us to kind of the next step in the evolution of the use of these Bio-Sep beads is with asking the question of whether or not we can use these same properties, that it's absorbed its surface, the high tendency for microorganisms to grow, in these beads and develop this into a treatment process. And that's exactly what we've done with the Bio-Sep ISBR. And I'd like to start with a typical diagram for what a typical ISBR would look like. And I'd like to walk you through the function of this particular device. So the central part of the ISBR is basically just a closed pack bed of Bio-Sep beads. And at either end of this closed pack bed is a connector here that is porous, and with a top section and a bottom section that provides hydraulic connection to groundwater in a groundwater monitoring well. This whole device is about 36 inches long, or about 1 meter long. It's made of 1.25 PVC, so it easily fits into a 2-inch well. Now, other features include, on the inside of the pack bed, down at the bottom here, is going to be a sparger where we can deliver, as you see on the top here, through this Teflon tubing, we can deliver a gas. That gas can be air under aerobic conditions, or if we want to operate anaerobically, that gas can be nitrogen. And as we sparge that air or nitrogen into the bottom of the pack bed section, it lowers the total density in the pack bed, and we set up a circulation pattern. You may be familiar with an airlift type of bioreactors. That's basically what we have here, where as we reduce the average density on the inside of the pack bed, groundwater will flow in through the bottom of the ISBR, flow into the pack bed, up the pack bed, and out the top. So basically, we're setting up in the well a circulation pattern that looks similar to the motion of the arrow that I'm showing here. So effectively, as these Bio-Sep beads become colonized with the indigenous degraders that are already present there in the aquifer, then as that groundwater circulates through that pack bed, then it comes in contact with a high concentration of these degraders, and that sets up the degradation process. Now, degradation occurs as I've just described it, but one of the most important features, I think, of the in situ bioreactor has to do with properties of the Bio-Sep beads. Bio-Sep beads do have a finite carrying capacity, which means that microorganisms will grow and multiply on the inside of the beads up to a point at which basically the available space is occupied by biomass. And at that point, then, growth does not stop, but growth continues. We've showed this many of times in laboratory bioreactor systems. And as that growth continues, then new microorganisms, new degraders that you've grown up inside the ISBR are released from the Bio-Sep beads and then are then carried out of the in situ bioreactor and released into the wellbore area. And some of the case studies I will show you or make available to you will demonstrate to you that these microorganisms now we've released into the wellbore area now have the capability of growing into the aquifer and following a concentration gradient. And in that way, we affect biodegradation not only in the wellbore, but outside of the wellbore as well. Now, one other thing I wanted to point out to you is there's a second tube up here at the top, and that the purpose of that tube is to introduce into the reactor, and it's introduced down by the sparger, so the sparger can provide mixing. Any kind of nutrient addition that you want to add to the ISBR. So for example, for petroleum hydrocarbons, we might want to introduce a source of nitrogen and phosphorus if we're using air. So we provide the electron acceptor as well as providing the required nitrogen and phosphorus nutrients that the organisms need. If we are operating anaerobically, we might be introducing an electron donor into the ISBR to make that electron donor available, as Brad was describing, to be fermented in the ISBR, produce hydrogen then to fuel things like reductive dechlorination. So either anaerobic or aerobic applications can be handled with the ISBR. It's a matter of determining whether we're going to use air or nitrogen. And by the way, if we use nitrogen, the nitrogen addition is provided through a compressed gas tank, but it's intermittent. We've got it on a controller so that nitrogen is provided periodically to produce that circulation in the bed, but it does not have to be added to the bioreactor continuously. So that's basically how the in situ bioreactor works. Now, of course, we do need some topside control. And that's what's shown in this box right here. This box will contain nutrient reservoirs and pumps. It'll contain, for aerobic applications, it'll contain an air pump and an airflow controller. And I'll show you one for anaerobic applications in a few minutes. So these are some of the applications that we have tried and or envisioned for the in situ bioreactor. Petroleum hydrocarbons, either under aerobic or anaerobic conditions. Under anaerobic conditions, we could provide through that nutrient line an alternative electron acceptor like nitrate, sulfate, etc. Under anaerobic conditions, we can also provide, again, as I said, electron donor to fuel reductive dechlorination. And then also fuel oxygenates like MTBE, TBA, or emerging contaminants like 1,4-dioxane. Now, I want to show you a couple of case studies. I don't have time to show you a complete set of case studies, but I'll also refer to that at the end of the talk so that you can get copies of the additional case studies as well. But the first one I want to talk about is an application of the Aerobic ISBR in a situation with low concentrations of hydrocarbons. This in particular was a fuel oil release that was impacting soil and groundwater below a private residence. And we installed a 2-inch monitoring well here where the in situ bioreactor was going to be deployed. We also installed some monitoring wells down a gradient. There was an existing sump that was also over here to the right here, and the withdrawal of groundwater from that sump was producing groundwater flow in that direction. So as I said, we installed the in situ bioreactor here to evaluate its performance, but we also installed biotraps in all of these monitoring wells as well as in the bioreactor well to monitor the microbiology. Now, we're going to be adding air, and we're going to be adding nutrients. So a perfectly relevant question is whether or not just adding air to the aquifer or just adding air and nutrients to the aquifer will result in stimulation of some of these BTEX hydrocarbons that have been mobilized in the groundwater. So to answer that question and to fully evaluate the effect of the bioreactor system and the effect of the Bio-Sep beads in the bioreactor system, we conducted a study in which for approximately the first 30 days of operation, we had the in situ bioreactor in place, but there was no Bio-Sep beads in the bioreactor. And for 30 days, we sparged the in situ bioreactor and circulated groundwater through the in situ bioreactor with that sparging. For the next 30 days, we added nutrient delivery. So we're adding now a source of nitrogen and phosphorus to the in situ bioreactor as well as sparging, but still there's no Bio-Sep bead. So for the first 60 days, there's no Bio-Sep. And then for after that 60-day period, we then withdrew the bioreactor system and filled it with Bio-Sep beads so that it was now completely operational, and we continued then to feed air and nutrient to the bioreactor, as I said, now with Bio-Sep beads. So I'd like to show you some results here. Again, these are results that come from the biotraps. These particular results are from the biotraps that are actually in the bioreactor well. And what we're looking at here are qPCR results, the process that or analysis that Dora was telling you about a few minutes ago. And specifically, we're looking at RNA. And the reason we're looking at RNA, as Dora indicated earlier, there are some types of genes that we find in DNA and bacteria that you will just find almost everywhere. And one of those examples are catabolic functional genes for hydrocarbon degradation. Hydrocarbon degraders are everywhere. But if you find the gene, that doesn't necessarily mean that gene is being expressed, as Dora was explaining. So the better picture of activity of hydrocarbon degradation comes from an analysis of RNA. If we find that gene in the messenger RNA molecule, it indicates to us then that that gene is actually being expressed. And if it's being expressed, it's being expressed for a function, and that is to produce an enzyme involved in the degradation process. So we have two of those functional genes on this particular display, and also a third qPCR result here. This is what we call EBAC, and basically that's just total true bacteria. So what we can see from this particular result is for the first 60 days of operation, when we were either just sparging the in situ bioreactor or we were sparging and adding nutrients, there was no stimulation of the expression of these catabolic functional genes for aerobic hydrocarbon degradation. It was only after that 60 days of operation, when the Bio-Sep beads were placed in the bioreactor, that we saw a stimulation in the expression of hydrocarbon degradation genes, these hydrocarbon catabolic genes. Again, these would not be expressed unless the organism was intending to produce those enzymes and use those in the degradation process. And as you can see here, for the next about 180 days or so, after the introduction of the Bio-Sep beads in the bioreactor, we continue to see the expression of these catabolic genes. And then, of course, we also saw the disappearance of the BTEX components, or a reduction to very low levels of these BTEX components, starting about the time the Bio-Sep beads were introduced. Now, the second case study that I want to tell you about is an anaerobic application. Here is the topside control for the anaerobic in situ bioreactor. As far as the downhole system is concerned, as I said earlier, the only difference is the injection of nitrogen into the in situ bioreactor and the injection of an electron donor. This is the control system here with the reservoirs for the electron donor, as well as a metering pump to meter that down to the in situ bioreactor. And this one of these rotometers here is simply the nitrogen flow rate to the in situ bioreactor. This particular case study was a particularly difficult one. It was a chlorinated solvent impacted site. It was fractured bedrock aquifer. Very deep groundwater impacts. We've had about a water column about 140 feet high. The geochemistry was relatively unfavorable for an anaerobic application, in that the DO was fairly low, but anytime there was a rain event, we got these spikes in the dissolved oxygen concentration. So what we did was to install an in situ bioreactor at a depth of about 30 feet. So there's about another 110 feet below the in situ bioreactor. A liquid carbon was used as an electron donor. My colleague Eric Reyes could probably tell us what that was, but basically it was a fermentable carbon source. We monitored the groundwater looking at contaminant concentrations and geochemistry. And then we use qPCR for the helicocoides, as well as some of the functional genes that code for enzymes involved in the reductive dechlorination process using biotraps. Long story short, I know this is a busy slide, but it kind of summarizes all of our results. After installing the in situ bioreactor, you see the TCE concentration is dropping, followed by an increase in DCE, which subsequently decreases with increases in concentration of vinyl chloride, as well as ethene. The bars indicate the qPCR results. The dark bar here is the taxonomic gene for the helicocoides, and the other bars here are for some of these functional genes involved in the reductive dechlorination of TCE and PCE. And as you can see, corresponding with the decrease in concentrations or the stimulation and the reductive dechlorination of the TCE and a little bit of PCE, there's a corresponding increase in the concentrations of the helicocoides as well as these functional genes. Now, what's really interesting too, if we look at the distribution of the helicocoides in this deep well prior to introduction of the in situ bioreactor and following introduction, as you can see, we've stimulated growth of the helicocoides through the entire depth of that well. If we look at those functional genes, we had one functional gene detected at a shallow depth prior to operating the ISBR, and then after operating the ISBR, we're now detecting those functional genes in high concentrations over the entire depth. Now, this has been a very short presentation with only 2 case studies. And I would like to, as Brad indicated earlier, I would like to invite you to, if you have an interest, to contact me. There is my email address at the bottom, or you can contact Brad directly. And I would like to provide you with an unabridged copy or version of this presentation. And there are 3 additional case studies there I would like you to see. One involving treatment of a site with 6 different bioreactors where the toluene concentrations were very, very high, 460 milligrams per liter. That's not a mistake, that's milligrams. Inhibitory concentrations of toluene, where we were able to reduce those concentrations very significantly. A case study where we paired the in situ bioreactor with an ISCO process for treating hydrocarbons. And also there's some discussion in there and some data describing the use of the in situ bioreactor to actually cultivate the helicocoides in the in situ bioreactor and then transfer that bioreactor to another well where there's not very efficient reductive dechlorination going on, resulting in the stimulation then of that reductive dechlorination. There's also more discussion of recommended applications as well as cost and some of the limitations of the ISBR. And one of those limitations I would like to tell you right now, and that is that the ISBR basically works by inducing that circulation process that we talked about. So the ISBR must be at all times fully immersed in groundwater. If the water table drops and you expose the top of the in situ bioreactor, then you're not going to have that circulation and the in situ bioreactor will cease to function. So we need to keep that in situ bioreactor completely covered with groundwater at all times, and if we do, it'll be fully functional. So with that, I will turn it back over to our host, and I think he's going to solicit questions. Thank you very much.
That's right. Thank you so much, Kerry. Dora and Kerry, just before we get into questions, I just want to extend my utmost appreciation for your time today and joining me on this webinar. Both presentations were were great, and I hope that everyone learned something new today. So let's dive into the questions, and the first one is, I believe, geared towards Dora, and I'll read it. And Kerry, obviously, if you feel like you need to jump in and have a rebuttal, please do so. Have you been able to look at co-oxidative chlorinated ethene pathways, for example, aerobic propane degradation with TCE co-metabolism?
We have looked at quite a few of the co-metabolic pathways. We have, for propane degradation, we can look at the propane monooxygenase gene to see if that's being stimulated. We have actually been doing a lot of work with John Wilson and Todd Wiedemeyer looking at some co-metabolic degradation for a startup project that they were involved in. They're going to be presenting this data at the Battelle conference. And one thing that was interesting is we found that the phenol hydroxylase gene tracked really well with some chlorinated ethene degradation in the studies that they did. And so we think that may be a really important gene for us to look at for co-metabolism. It seemed to track better with some of these lab studies that they did and field studies, even more so than like the SMMO gene that we traditionally have been looking at. So yes, we have done quite a bit of that, and I think that's an evolving and emerging field, and I do feel personally like it's something that we underestimate the amount that reaction is actually happening in the environment and how important of a component that might be at sites. So I don't know if there's anything else that you can think that we want to add on that, Kerry.
Well, just that we haven't done this yet, but we are currently quoting an application where we are looking at feeding propane to an in situ bioreactor specifically for the purpose of evaluating these co-metabolic pathways in situ that you were just describing, Dora.
Next question is for Kerry. What is the position of the in situ bioreactor relative to the screen interval of the well in which it's inserted?
Well, it depends on how much information that you have about the contaminant interval. If you have that kind of information, then you're going to try to position the ISBR as close to that contaminated interval as possible. If you don't have that information, we suggest that the end user position the in situ bioreactor at the midpoint of the screened interval.
Any estimation on the observed radius of influence with these bioreactors?
Good, great question. I can tell you that one of the case studies that is in the unabridged version of this presentation that I invite you to request, the one involving toluene, where we had a fairly large plume of toluene that was fairly shallow, and we had 6 of these in situ bioreactors that were deployed at this particular site. We were using stable isotope probing, the technique that Dora was describing a few minutes ago, to actually document toluene biodegradation and how we were stimulating toluene biodegradation at the site. The aquifer matrix here was a fairly silty type of material, yet we were able to show that we could significantly stimulate toluene biodegradation in a monitoring well that was located 10 meters away from the closest ISBR. It took about 3 months to do it, but we had significant increases in a number of different monitoring wells, but our star pupil was the one that was 10 meters away. So the radius of influence is going to depend a lot on the aquifer matrix, but it's also going to depend upon the growth characteristics of the degraders in that particular aquifer matrix. And if they can make it 10 meters in 3 months through a silty aquifer, then I think we're going to have significantly high probability of large radii of influence in most of these aquifers.
Great. Next question is for both of you. Looking at the chlorinated solvent site, I believe that Kerry was talking about during his presentation, did you monitor the RNA to demonstrate that the genes were expressed?
We did not, and Dora, maybe you would like to explain the DNA versus RNA decision on the helicocoides?
Right, so we did quite a bit of research on this actually, and what we found, because the helicocoides don't have a lot of metabolic capabilities like other organisms, you know, when we're talking about petroleum hydrocarbon degradation, these bacteria like Pseudomonas have a lot of different metabolic capabilities. So we need to know, are they expressing the one we're interested in at that point? For the helicocoides, we did a lot of studies looking at DNA and RNA and the correlation, and what we found is if your DNA is high, your RNA was always high as well. It may not be the exact same numbers, but these organisms are not typically going to be present in the aquifer unless they're active, not in higher concentrations above that 10 to the 4 cells per milliliter. If you're up at 10 to the 5, 10 to the 6 at the helicocoides, there's a good portion of those that are actually doing something in that environment. We just don't see them high without that expression because they're not going to be able to survive and make additional biomass unless they're doing the functions we're interested in, because that's the only function that they do to survive. So I don't know if there's anything else you think we should add to that, Kerry.
No, I think that's right on the money. As you were saying, as far as hydrocarbon degraders are concerned, these same organisms that do hydrocarbon degradation have a lot of other capabilities, and their presence there really doesn't tell us much about what it is they're doing for a living, unless we look at the RNA. And as you were explaining, that is not the case with the helicocoides.
All right. I think we got time for one last question. And again, this is another one that was asked more than once. And that is, and Kerry, this is directed at you. What kind of permit requirements or sort of regulatory permission have you had to go through to deploy the in situ bioreactors?
Really only one, and that is that if you are in a regulatory climate where a permit is required to introduce a remediation amendment to the aquifer, then this represents the introduction of an amendment that's being fed to the ISBR, and that's the only permit that we've seen that you're required. All
right. Well, that's going to wrap us up. We're at the 1 o'clock hour. So I'm going to bid everyone a farewell. Kerry and Dora, again, I really appreciate your time. Great presentations. And if anyone has any questions that didn't get answered, please feel free to either email Kerry, Dora, or myself directly, and we'll answer those questions as we receive them. Have a great afternoon, everyone. Take care.
Thank you.
Thank you.